Hello, today we are going to discuss few things about neuromuscular junction or transmission. So, this is a nerve terminal, this is an axon terminal, okay, and these inside the small dots are the synaptic vesicles. We'll discuss. And this is a muscle membrane and muscle terminal, and this whole curved shape is known as synaptic trough or synaptic gutter. This is invaginated membrane of the muscle, and it is forming these clefts. This is known as subneural clefts to increase the surface area of this trough, and the space between the axon terminal and this synaptic clefts, this space is known as synaptic clefts. This space is known as synaptic clefts. Okay, and this is approximately 20 to 30 nanometer wide, and it is generally asked. As you can see in the axon terminal, there are a lot of mitochondria. These are the mitochondria which is generally there to supply, as I know, mitochondria is the powerhouse of the cell. So it supplies ATP for the synthesis of acetylcholine. Here in the axon terminal only, the acetylcholine is synthesized, and these vesicles which you are seeing here, in which acetylcholine is stored, these vesicles come from the cell body, because in cell body there are Golgi bodies, and in the Golgi bodies these vesicles are created, and then it is transported till the terminal, and acetylcholine is formed here only. In these spaces, this synaptic clefts, there is an enzyme known as acetylcholine esterase, which is used to break down the acetylcholine into acetic acid and choline. So when the action potential impulses or nerve impulses reaches the axon terminal, then in the terminal 125 vesicles of acetylcholine approximately are released into the synaptic space. How it is secreted? On the axon terminal there are the calcium channels. So you can see there is a dense part, and there are the calcium channels, and these are the vesicles. So as the action potential reaches the axon terminal, what happens? These calcium channels are opened. Okay, these calcium channels are voltage gated calcium channels. So when the voltage, that is action potential, is reached here, then the calcium channels open, and from here the synaptic this space, the calcium channels go inside, and when the calcium channels go inside, it activates a protein, that is calmodulin dependent protein kinase, which phosphorylates the protein on acetylcholine. So what happens with the phosphorylation of the protein on acetylcholine? Acetylcholine is released from the vesicle. Okay, so acetylcholine it frees the acetylcholine vesicles. These acetylcholine earlier were held by the cytoskeleton of this region, and when they are freed from the proteins, then they are freed from the cytoskeleton, and these acetylcholine move to the active zone of the presynaptic membrane. Okay, and then they are released by, you can see the process, exocytosis. Okay, it is also known that effective stimulus for causing acetylcholine release from the vesicles is the entry of the calcium ions. So when the calcium ion enters, that is the main stimulus for the release of the acetylcholine. But for the calcium ions entry, the action potential is needed. So when the acetylcholine, this is a subneural cleft. Okay, so when the acetylcholine comes here, there are the acetylcholine receptors over here. So when the acetylcholine reaches and joins the acetylcholine receptors, what happens? These receptors get opened. So this is acetylcholine, okay, and these receptors get open. And this is not specific to sodium ions only. It can enter sodium ions. It can allow the diffusion of potassium ions outside. But as we know, inside the membrane is already it's negatively charged. Okay, so it allows the movement of the sodium ions inside instead of the potassium outside, because of the strong negative charges in the mouth of the channel. You can see it repels the negative charges, and it allows the sodium ions to come inside. So as it comes inside, the more and more voltage activated sodium channels also open, and then more sodium is coming inside, and this creates positive potential change inside the muscle membrane. That is known as end plate potential. and this end plate potential initiates action potential that spreads along the muscle membrane, and causes a muscle contraction. Here in this space there were acetylcholinesterase. So when the action potential reaches the muscle membrane, then acetylcholinesterase destroys the acetylcholine into acetic acid and choline to avoid the re-excitation of the muscle membrane. These acetylcholinesterase are inactivated by nerve gas, that is diisopropyl phosphate, fluorophosphate. That is the acetylcholine. This esterase is destroyed, so more acetylcholine is there, which causes muscle spasm, which causes re-excitation of the muscle, and muscle spasms are caused. The nerve gas, the isopropyl fluorophosphate, very lethal. There is one more disease, myasthenia gravis, that is autoimmune disease. What happens? These are the acetylcholine receptors on the subneural clefts. So what happens? There are antibodies which our own antibodies which attack these receptors, acetylcholine receptors. So there are less acetylcholine. Okay. Then what we do to resolve this problem? We destroy the acetylcholine esterase. Okay, so when we destroy acetylcholine esterase, less acetylcholine less acetylcholine will be breaking. So more acetylcholine will enter. Okay, so it will accumulate here, and the normal amount of acetylcholine accumulate in the synaptic space. So within few minutes, it begin to function almost normally. So this was about neuromuscular transmission. Thank you.